Okay, it's my pleasure to introduce Dave Hutchins by phone. He apologizes that he can't be with us today for family reasons. Dave is going to give a lecture on biogeochemical feedbacks. And with no further ado, this is Dave from USC.
Thank you, Heather. Good morning, everyone. Sorry I have to give this lecture as a voice over the phone, but as Heather probably told you, my wife is expecting a baby pretty much any day now, so it wasn't a really good time for me to come out to Whitstable. But thanks for being patient with me, and I will follow along as best I can from here. And probably, because I can't see you, probably best to hold questions till the end because I can't see you or call on you. Do you think that's the best way to do it, Heather?
Yes, that would be good.
All right. Well, let's go ahead and get started then. I assume the first slide is up there, the title.
Yes.
What Heather and Scott asked me to talk about is the potential effects of ocean acidification on marine biogeochemical cycles and the different ways that have been used and could be used to investigate effects of OA on biogeochemistry. Because most of the biogeochemical cycles in the ocean are mediated by microbes, that is phytoplankton or prokaryotes like eubacteria and archaea, that's primarily what I'm going to be talking about today. I'm not going to address animals and so forth very much. You're going to be getting a lot more of that from other people, other instructors in the course. I'm going to talk mostly about microbially mediated nutrient cycles today. And for each, I'm going to go through the carbon cycle, and then the major nutrient cycles, nitrogen, silicon, and phosphorus, and then I'm also going to talk about trace metals. And for each of these topics, I'm going to show you some of the, just a little synopsis or a few snapshots of some of the work that's been done in these areas. And then I'm going to talk about, at the end of each section, the types of methods people use to make biogeochemical measurements. I'm not going to focus on things like, ways to measure gene expression of biogeochemically important genes and so forth, even though those types of approaches are really valuable in biogeochemistry. Again, I think you're going to be getting more of that from the other instructors. So I'm going to focus mostly on strictly biogeochemical methods. How do you follow where the elements are? So let's go to the next slide. I guess the first question you can ask is, why should we even expect ocean acidification to affect marine biogeochemical cycles? And the simple answer to that is on the next slide. And that's because, of course, as oceanographers, we recognize that there is no such thing as an independent biogeochemical cycle, is that the cycle of carbon, which is being perturbed by all this CO2 entering the ocean, is inextricably linked to the cycles of all the other elements. And I've just put up a little cartoon of the elemental ratios of phytoplankton, which of course is one of the basic fundamental concepts of biological oceanography dating back to Redfield. And you can see that it certainly makes sense that if the cycle of carbon changes there on the left, that, for instance, the cycle of the other major nutrients, nitrogen, silicon, and phosphorus, also are likely to be affected in different ways, just looking at it from the phytoplankton elemental ratio point of view. And of course, there's a lot of other places where these different biogeochemical cycles intersect in the ocean as well. Next slide, please. And this is one that still has the animation in it. Sorry about that. You're going to have to hit it more than once. So if we look at the Redfield ratio, which is 106 carbons to 16 nitrogens to 1 phosphorus, and you can put silicon in there, even though Redfield didn't talk about that if you're talking about diatoms. And then click again, Heather, sorry about that. You can see that there have been studies that take this kind of elemental ratio concept and extend it beyond carbon and the major nutrients. So there's a paper by Ho et al. In which they looked at the elemental composition of phytoplankton down to trace metals, and this is the formulation they came up with. There's another paper by Price in 2005 in which he also extended the idea of Redfield ratios to iron, and you can see the iron replete and iron limited ratios that he came up with. So my point being, again, if the cycle of carbon is changing in the ocean, not only major nutrient cycles are going to be affected, but also those of its bioactive trace elements like iron. And I'll talk about that at the end of my lecture. Next slide, please. So let's talk about carbon first, since it's the most important part of this course. A lot of times, I think when we mention ocean acidification, we have this idea that it is an unmitigated stressor for marine organisms. And frankly, that's probably true for most animals. It's hard to envision how an animal could benefit from higher CO2 or lower pH. But that's not the case for many microbes, and this is because a lot of the microbes that are important in ocean biogeochemistry are autotrophs. And so from their point of view, you could call it ocean acidification, or you could just as easily call it carbon dioxide enrichment or fertilization. So because they use CO2 to obtain carbon, there is a potential for them to actually benefit from higher CO2 levels. And this includes potentially some phytoplankton and also chemoautotrophic bacteria, both of which I'll be talking about today. The responses of these autotrophs to higher CO2 depend partly on carbon concentrating mechanisms. I'm not going to talk a lot about CCMs today, but basically what they are is they are various biochemical ways that cells have evolved in order to concentrate CO2 around the ribisco enzyme, which fixes carbon. And these include things like carbonic anhydrases. So I'll come back to these a little bit later in the lecture. And the general idea is that those groups that have the least efficient CCMs, that is, they're the least good at concentrating CO2 for carbon fixation, may be the ones that benefit the most from increasing PCO2. In other words, as CO2 goes up in the seawater, CCMs may become less advantageous, is another way to say that. If you think about calcifying autotrophs, that is coccolithophorids, which you'll hear a lot about in this course, it's certainly possible that they're going to benefit, their photosynthesis is going to benefit from higher CO2, while at the same time their calcification could be reduced. So the effects of CO2 on coccolithophorids can be a bit schizophrenic in that they may actually benefit in their organic carbon production may benefit, while their calcification could potentially be reduced. For both carbon fixation and calcification, a theme that you'll see throughout my talk, and I think that's really important to come across, is that we need to think beyond just acidification and elevated CO2. There's a lot of things changing with global change in the ocean. And many of the data I'm going to show you, the work that my group has done and others, is showing that interactions of acidification with things like rising temperature or changing light or changing nutrients can be at least as important as the effects of CO2 alone, and in many cases more so. I believe that you can't make predictions based on just ocean acidification manipulations. If you really want to know how it's going to change in the future, you need to bring the rest of the global change picture into it. So let's go on and I'll show you some examples of the response of carbon biogeochemistry to ocean acidification. Next slide shows some work that my lab published looking at harmful bloom flagellates and how their photosynthesis responds to higher CO2 and to temperature. And what I've got here is a PE curve. So this is basically the carbon fixation rates of these two groups plotted versus light intensity on the x-axis. It's a very standard physiological method in phytoplankton biology. And this experiment is a 4-treatment matrix, and you'll see a lot of these in the examples I give. So what we did is we have a control that's at present day PCO2 and present day temperature. That's the black circles. The open circles are high CO2, but at present day temperature. The black triangles are at elevated temperature, but present day PCO2. And then the open triangle is what we call the combined, or the greenhouse, we like to call it. So this is the high CO2, high temperature treatment, which is really what's going to happen in the future. And if we take a look at the top panel, this is the harmful flagellate heterosigma. It's a rhaphidophite. And what you can see right away is that photosynthesis increases with light and then levels off at a maximum rate. And that in the control, the high CO2 and the high temperature, that maximum rate is very similar and not significantly different. However, if you increase CO2 and temperature together, the open triangles, there's a much higher maximum photosynthetic rate. And so there's kind of a synergism between acidification and warming temperatures that allows this harmful flagellate to photosynthesize faster relative to the light it's getting. The bottom panel is a harmful dinoflagellate, Proacentrum, and you can see it has a different pattern. For this species, it benefits from the higher CO2, those open symbols, whereas the two lower temperature treatments, the solid symbols, have about the same maximum photosynthetic rate. So this has, for Proacentrum, we see less effective temperature, and we see that they are benefiting from CO2, just as I mentioned a few moments ago. This is a good example of what I would call carbon enrichment or carbon fertilization. Next slide, please. So this is a natural community experiment we did up in the Bering Sea. It's the same type of an experiment, a temperature CO2 matrix of photosynthesis versus a radiance curve. And basically what you can see is the same thing we saw with heterosigma cultures is happening with this natural community up in the Bering Sea, in that the maximum photosynthetic rate is increased by the combination of high CO2 and high temperature together. But either one alone are not significantly different from the control, the lower values. Next slide, please. This is data from a paper that we just published in which we did a similar CO2 temperature matrix experiment in the North Atlantic spring bloom. And this illustrates some of the principles I was talking about, coccolithophorid photosynthesis and calcification. So in this set of graphs, black is the ambient control, red is the high CO2, green is the high temperature, and yellow is the combined high CO2 and temperature or greenhouse treatment. If you look at the top left panel, this shows the abundance of coccolithophores in our incubation experiment. And you can see really interestingly, there's no change in the number of coccolithophores when you increase CO2 or temperature alone relative to the control. But when you increase them together, you get a coccolithophore bloom. The coccolithophores simply love the greenhouse effect, the yellow bar there. And if you move over to those three right panels, this shows you the production of organic carbon, in particular inorganic carbon, in this same incubation. The top panel, the POC production, you can see there really was almost no effect of CO2, but the two high temperature treatments, the green and yellow bars, fixed carbon faster on the right. If you look at the production of calcite, PAC production, you see a different pattern in the next panel down. There was no effect of either CO2 or temperature increases alone on the production of PIC. But when we increase them together, we see much lower rates of PIC production in the greenhouse treatment. And then finally, if you look at the bottom panel, the pick-pock ratio, that is, how calcified is this community, you can see by far the least calcification is happening in that yellow greenhouse treatment, which sort of paradoxically is the one that had the coccolithophore bloom. So this illustrates what I was saying in that, in this case, what we had was the coccolithophores benefited from the high temperature, high CO2 treatments, but they were very, very lightly calcified cells. So we had a bloom of coccolithophores, but not much calcite being produced. Go to the next slide, please. And this is illustrates, taking it one step further, this is a lab study of the coccolithophore E. hux, in which we did a 3-temperature matrix. So what we have is our 4 treatments: ambient, high CO2, high temperature, and greenhouse. And each of these was grown at high light, the open bars, and low light, the black bars. And what you can see is in this strain of E. hux, there is no effect of either temperature or CO2 on calcification, the POC ratio there, at low light. The black bars, when they're light limited, there was no effect of either of those variables. If you look at the open bars, you can see that when we increased light to be supersaturating, 400 microeinsteins, it decreased calcification in all treatments. But then, within the treatments, if you compare the ambient and the high CO2, you can see CO2 did decrease the calcification of the cells, and the same between the high temperature and the greenhouse. So our conclusion from this is that the primary control on calcification in this E. hux strain was light intensity. However, once you have a saturating light level, CO2 exerted a secondary effect and further decreased the calcification of the cells. And then finally, for a final example in the next slide, this is data from a paper by Ulf Rivesel that he published a couple of years ago from one of his Bergen mesocosm experiments, in which they showed so-called carbon overconsumption at high CO2. So this plots the utilization of nitrate in the mesocosms on the y-axis versus the drawdown of DIC on the x-axis. And the slopes of those lines change with CO2. So at the lowest CO2, the 350 ppm, you can see the C to N drawdown ratio was 6. But as they increase the CO2 up to 1,050 ppm, the C to N ratio increased up to 8.8. In other words, the phytoplankton in there were consuming more DIC at high CO2 relative to the amount of nutrients that they were using. And I'll come back to this carbon overconsumption in a few minutes. Next slide, please. So to talk for a moment about how you make measurements like the ones I just showed you, I am not going to talk about DIC drawdown like Ulf measured in his mesocosms. I think you're going to get a lot of that from the other instructors. The same with calcification and organic carbon fixation using C14. I think Deborah and Barney are probably going to show you a lot about that and some of the other instructors. So I'm not going to cover that. But just to look at bulk changes in POC, the typical way we do that is we use a CHN analyzer, which is actually a very simple gas chromatograph that combusts the sample and converts all the organic carbon to CO2 and then measures it. This allows you to get total organic carbon in these samples. Of course, if you take duplicate samples and acidify them, by difference, you can get an estimate of inorganic carbon or calcite and aragonite as well. And the nice thing about CHN analyses is they also give you particulate organic nitrogen at the same time. And I've given you one of the original references for this method. You can also get mass balances of POC and PON using a mass spec, but that's a lot of work to get that. It's much easier to use a CHN unless you need to look at stable isotopes. Most people use a CHN analyzer, although you do get the same information from a mass spectrometer. For our final methods on carbon fixation measurements, the PE curves, photosynthesis versus irradiance, you basically need a simple instrument called a photosynthetron, which produces a gradient of light levels. The one we have is made by CHPT Industries. If you're interested in one, email me. I'll put you in touch with them. It uses fiber optic light to make a range of light levels from about 0 to 1,000 microeinsteins. You spike your phytoplankton samples with C14 and incubate them for a very short half-hour incubation to get an estimate of how gross photosynthesis responds to light. And I've given you some references for that method as well. So let's move on to the next slide and talk for a few minutes about nitrogen. The marine nitrogen cycle is really complex, as probably most of you know. It spans 8 oxidation states from ammonium at -3 to nitrate at +5. And I diagrammed some of the major pathways. The ones I'm going to concentrate on in this lecture today are denitrification, N2 fixation, and nitrification, the ones that are highlighted in green, red, and white. Anammox and assimilation of nitrogen are important pathways, but the reason I'm not talking about them is almost no work has been done that I know of to look at effects of acidification on these processes. It needs to be done. Let's go to the next slide. I'm going to start off by talking about nitrogen fixation, the red arrow there, which converts N2 gas to the level of ammonium. Next slide. Biological nitrogen fixation is the way that fixed nitrogen enters the ocean mainly, although certainly there's an anthropogenic input to consider these days. But naturally, that is the main way that fixed nitrogen is produced in the ocean. The major groups that carry out this are all prokaryotes. Cyanobacteria are very prominent. The colonial cyanobacterium trichodesmium is well recognized as a major source of fixed nitrogen. There's a picture of it there on that slide. There's also a lot of unicellular nitrogen fixers that we've more recently become aware of. I've listed Crocosphaera, although there's a number of others that are being found. There's also symbiotic relationships between nitrogen fixers and diatoms and zooplankton that can be important. And there's also probably free-living eubacteria and archaea that can also fix nitrogen. But it's a purely prokaryotic function. Most of the nitrogen fixation is thought to occur in the oligotrophic gyres and the subtropics and tropics, although there's now more being found in temperate regimes, and that may change a bit, but we think that it's most prominent as a biogeochemical pathway in the gyres. Because nitrogen fixers won't be limited by nitrogen, they can obtain essentially unlimited N2 gas. They're probably often limited by the availability of either phosphorus or iron, and there's quite a literature on that. Next slide. This simply shows new production supported by nitrogen fixation over the Earth's surface. It's a model simulation of new production, and you can see how important nitrogen fixation is in those tropical and subtropical regimes. Next slide. So I guess the question that we want to talk about here is how is nitrogen fixation going to change in the acidified ocean? And there are a few papers out now that suggest it's going to increase rather dramatically. There's 4 papers that have used cultures of trichodesmium and 1 that has used cultured Crocosphaera, and in all cases they have reported large increases in nitrogen fixation rates as PCO2 goes up from present day to future levels. Next slide. I've plotted some of these literature results here. And what I plotted is the % increase in nitrogen fixation rates reported from present day PCO2 to future PCO2, mostly 750 ppm, although a couple studies use different values, and I've given you the details below, but don't worry about those right now. I guess the main take-home message is, between all these studies, they've reported increases in nitrogen fixation rates over, say, the next 100 years that range from 35% up to as much as 120% in trichodesmium and in Crocospira. And they all have fairly consistent results in that respect. Next slide. Those are all culture studies. What's lacking in the literature is field evidence. And here is a little bit of field evidence here, but we need a lot more. We incubated natural trichodesmium colonies from off of Florida for just a few hours at 380 and 750 ppm. And in all three cases, nitrogen fixation rates increased between 6 and 41% within a few hours of increasing the CO2. Suggesting that what we see in cultures can happen in the ocean as well, and that CO2 is approximate limitation on nitrogen fixation, at least in this case, because they respond very quickly to higher CO2 levels with higher nitrogen fixation rates. Next slide. This is simply the projected future trends in global nitrogen fixation based on our culture studies. So I plotted the expected increase in CO2 in green there on the right axis, and in red and blue are the minimum and maximum increases in nitrogen fixation that we would predict based on our lab experiments. And if our lab experiments are predictive, that's a big if. But if they are, global nitrogen fixation by trichodesmium could increase from a present value of about 60 billion kilograms per year up to, about a minimum of 85 billion and a maximum of about 100 billion kilograms per year, simply over the next century. And then, according to our lab work, it levels out after that. But we certainly need a lot more work to understand the response of nitrogen fixation to changing CO2. And of course, this also depends on the availability of other limiting factors like phosphorus, iron, and light. So take this graph with a grain of salt. This is a projection, but it's not likely to become reality. Go to the next slide. This is, shows you the interaction between the CO2 effect on nitrogen fixation and phosphorus limitation in trichodesmium. What we did is we grew our 4 treatments on the bottom: control, high temp, high CO2, and greenhouse. And we grew them in the top panel in P-replete cultures and in the bottom panel in severely P-limited cultures. And the interesting thing is, if you look at the top panel, CO2 on the 2 right-hand bars increases nitrogen fixation rates in P-replete cells, just as I've been talking about. But interesting enough, even when they're severely P-limited in the bottom panel, it still increases nitrogen fixation rates by about the same amount, relative amount, I should say. If you look at the 2 axes of those x-axes, you'll see that the P-replete cells are fixing a lot more nitrogen, of course, than the P-limited ones. But in both cases, adding CO2 increases nitrogen fixation rates, even when they're limited by phosphorus. And this makes us think that you can add either phosphorus or CO2 to increase nitrogen fixation rates. In other words, they're co-limited by both of these limiting factors. Next slide. If we talk about the relationship between nitrogen fixation and iron, you can see that that it's a different story. This is for Crocospira now from the Fu et al. Paper. And what we did is we grew Crocospira, the unicellular nitrogen-fixing cyanobacterium, at 3 PCO2 levels down there on the x-axis and measured nitrogen fixation rates. And we did this in iron-replete and iron-limited cultures. And if you look at the top panel, you can see that just like trichodesmium, when they have enough iron, nitrogen fixation rates step up very nicely in a very linear fashion as CO2 increases. However, we see a different story for iron limitation than we saw for phosphorus limitation in trichodesmium. If you look at the bottom panel, once you iron limit the cells, the CO2 effect disappears. So they are not co-limited by iron and CO2. In this case, they are primarily limited by iron, and you have to relieve iron limitation before CO2 can have an effect. So go on to the next slide. This suggests that the two nutrients, iron and phosphorus, interact very differently with nitrogen fixation and CO2 interactions in these cyanobacteria. Iron is a kind of a traditional Liebig limitation in that iron limitation trumps CO2, so to speak. It cancels out the effect of CO2. And you have to give them iron before they can respond to higher CO2. On the other hand, P limitation does not cancel out the CO2 effect. Effectively, the trichodesmium cells we looked at were co-limited by both phosphorus and CO2, suggesting that CO2 could increase nitrogen fixation rates in P-limited parts of the ocean, but perhaps not in iron-limited parts of the ocean. But again, there's a need to go and do a lot more work to understand how the CO2 effect on this part of the nitrogen cycle is going to interact with other limiting factors. Next slide. I don't want to give you the impression that every nitrogen fixer is going to benefit from high CO2. There's now a little bit of evidence of at least one species that doesn't like higher CO2. Excuse me. This is a paper that was published by Zerny et al. This year in Biogeosciences. And what they showed is that the harmful bloom cyanobacterium, nitrogen-fixing nodularia from the Baltic Sea, their nitrogen fixation rates on the bottom panel and their growth rates on the top panel actually decrease as CO2 goes up rather than increasing as we saw for trichodesmium and Crocosphaera. So I think it's important that we look at a lot more nitrogen-fixing taxa before we're able to make really broad generalizations. Next slide. Just to talk to you for a moment about the types of measurements we use for nitrogen fixation. There's two generally applied methods. The first one uses gas chromatography, and it takes advantage of the fact that the nitrogenase enzyme doesn't really discriminate between the triple bonded N2 molecule and the triple bonds between carbons in the acetylene molecule. So you can use acetylene for an analog, and you can measure the rate at which it's reduced to ethylene, and using a conversion factor, you can back calculate the rates of nitrogen fixation. It's a quick and easy method to measure nitrogen fixation that gives you near real-time results. The second method is using N15 labeled N2 coupled with analyses by mass spectrometry. Of course, this has the advantage in that it uses the real substrate for nitrogen fixers. However, it's a lot more involved. You need an expensive mass spec, and you don't get real-time results. You have to wait and bring them back to the lab and analyze your samples later. But it has a lot of advantages over gas chromatography. In my lab, and the work we've been doing with our collaborators, we use both of these methods together. The reason for that, other than getting real-time results from GC, is that the acetylene reduction method gives you an estimate of gross nitrogen fixation, whereas the N15 method gives you an estimate of net nitrogen fixation, because you're only measuring what stays in the cells, not anything that they release. And it's a useful thing to know how much of the nitrogen that's fixed is released as DON or ammonium under these various experimental conditions. So they give you a little bit different information, and it is worthwhile to do both analyses if you can do that. So let's move on from nitrogen fixation and look at another part of the nitrogen cycle. Next slide. This green arrow is denitrification. Just like nitrogen fixation is the primary source of fixed nitrogen in the ocean, denitrification along with anammox is the way that fixed nitrogen is lost and removed from the ocean. And that involves the reduction from nitrate through a series of intermediates as far as N2 gas, thus completing the nitrogen cycle. Next slide, please. Just to summarize denitrification for those of you who don't know much about it, denitrifiers are very diverse phylogenetically. There's a lot of bugs that can do this, including eubacteria, archaea, and even some eukaryotes like fungi. The canonical denitrification is a purely anaerobic process, although many of these microorganisms are facultative anaerobes. And they're also heterotrophs. They're not autotrophs, at least in classical denitrification. So they're using organic carbon as a carbon source. What they're doing is using the oxidized nitrogen compounds as terminal respiratory electron acceptors. And so they reduce it through a series of intermediates, as I showed you, some often as far as dinitrogen gas. Denitrification only occurs in suboxic areas. The most important ones are the large areas of the ocean that have underlying suboxic zones like Peru, upwelling, the one off Central America, in the Pacific, and the Arabian Sea. Then denitrification, excuse me, also happens in suboxic and anoxic sediments all over the ocean. As I mentioned, along with anammox, denitrification is how fixed nitrogen leaves the ocean. Now, if we ask how denitrification is going to respond to ocean acidification, the easy answer would be, well, maybe it's not. And the reason that you might expect that is because these suboxic zones where denitrification happens already have extremely elevated CO2 concentrations, and they already have typically very low pH values. So the relatively minor, by comparison, changes that we're going to be making to the ocean pH from acidification may not have a huge effect directly on denitrifiers. At least that's the reasoning, although that has not yet been shown rigorously, but that's what you might expect. However, let's go on to the next slide. There is some idea that they are going to respond indirectly to ocean acidification and other global change processes. Again, this is that carbon overconsumption slide from Wolf's work in the mesocosm experiment. And their idea is that as CO2 goes up, phytoplankton will produce more organic carbon relative to the nutrients they use. When these particles sink out of the euphotic zone and decay, they're going to release this, and that will increase the amount of bacterial respiration in the ocean, and this carbon overconsumption could greatly decrease oxygen levels in the underlying water columns, thus expanding suboxic zones and potentially increasing denitrification all over the world. Next slide. This is a model that Oshleys et al. Built based on that carbon overconsumption model of ribosels. And what they did is they ran it out to the year 2100, and using this carbon overconsumption model, they predict on the top panel an increase in the suboxic water volume of the ocean by up to 50% over the next 100 years. A huge increase in these big water column suboxic zones. And of course, if you have more habitat, more suboxic areas, you're going to get more denitrification. So if you look at the bottom panel, they also predict about a 50% increase in denitrification over the next 100 years based on this high C to N production model that Wolf's experimental results suggested. I'm not sure that this is what's really going to happen. I think that the jury is still a bit out on this carbon overconsumption. I'll show you in a few minutes that I think there's evidence both ways, both for and against it. But it's an interesting and a little bit scary prospect that we could increase the suboxic water in the ocean by 50% over the next 100 years due simply to this carbon overconsumption and high CO2. Next slide. Even if you discount this carbon overconsumption, we still think denitrification is going to increase. And this is a couple of models that have looked at the amount of hypoxic or suboxic water as a function of warming and stratification. And so the idea is that as the ocean becomes warmer, surface ocean will, oxygen levels will decrease simply because warm seawater holds less oxygen than colder seawater. And so oxygen levels will go down at the surface. The oxygen levels in the deeper ocean may go down because of increased stratification and less ventilation. And so you can see these two models that I put here both have predicted large increases in suboxic areas in the ocean just based on warming and stratification without even considering the effects of changing CO2 and acidification. Next slide. So how do you measure denitrification? Well, it's not easy. You have to do everything in an anaerobic incubation if you're doing experiments, but there are ways that don't require incubations. First one is simply based on stoichiometry. So you can calculate how much nitrate a certain parcel of water should have, and then if the amount that's in there is less than that, you attribute this to denitrification. This is the so-called N-star approach that Curtis Deutsch and Gruber and Sarmiento have used, in which they use the phosphate concentration of a water parcel to calculate the expected nitrate concentration using Redfield. And a positive N-star would indicate that you're getting nitrogen fixation in that region, and a negative N-star would indicate that denitrification is happening. So this is kind of a broad geochemical approach. Secondly, you can also use a very targeted, focused biochemical approach. So you can measure the enzymatic activity of denitrifiers' electron transport systems using an artificial electron receptor tetrazolium that changes color when it's reduced. So it's a fairly simple colorimetric measurement that gives you an idea of how active denitrifiers are under various experimental conditions. And then finally, just like for nitrogen fixation, you can use stable isotope of nitrogen, N15, but in this case you would add N15 nitrate, and then you would measure the production of 29N2 using mass spectrometry, and Al Duvall has done that. And if you're interested in these methods, you can read the paper that he published in the new version of Nitrogen in the Marine Environment. Let's move on to the next slide. For the finish of my, excuse me, of my nitrification talk, or my nitrogen cycle section of this lecture, I'm going to talk about nitrification, the white arrows here, whereby ammonia is oxidized first to nitrite and then ultimately to nitrate. This process, you can think of it links the input process from nitrogen fixation with the output process from denitrification. So it closes the loop on the marine nitrogen cycle, which is why it's a very important process to look at. Next slide. Nitrification, as I said, it consists of the oxidation of ammonium, first to nitrite and then to nitrate. Classical nitrification is a purely aerobic process. There are anaerobic variants, but it's debated how important they are and where they occur. So we'll focus on the big one, which is aerobic nitrification, happens in the water column. And the prokaryotes are both eubacteria and archaea that can do this, are chemoautotrophs. So just like phytoplankton are using electrons from water, splitting water using sunlight to fix CO2, these prokaryotes are using electrons obtained from these reduced nitrogen compounds to fix CO2. They're autotrophs. We now know that a large fraction of global nitrification happens in fairly shallow waters. It tends to be inhibited by light, but nitrification maximum is typically right below the euphotic zone, making it likely that nitrification is going to be affected by ocean acidification in the fairly near term as CO2 continues to penetrate down below the euphotic zone and into the thermocline. To look at the eubacteria that do this process, the genus that oxidizes ammonia or ammonium is Nitrosomonas. The one that oxidizes nitrite is Nitrobacter. And one reason they could be affected by increasing CO2 is they both fix carbon using the Calvin cycle, just like eukaryotic phytoplankton and prokaryotic phytoplankton do. And since many phytoplankton are known to be potentially limited by CO2 that use these enzymes, it's certainly possible that nitrifiers could be CO2 limited as well. As far as I can tell, looking in the literature, almost nothing is known about carbon concentrating mechanisms in chemoautotrophic nitrifiers, so maybe that's a fertile area for investigation. What we do know is that the first step that's mediated by Nitrosomonas, the oxidation of ammonium, depends on the ammonia-ammonium buffer system in seawater. This buffer system has a pKa of about 9.19 at 25 degrees. And over the next century, if you do the calculation, the fraction of ammonia, which is about 6% right now, is going to drop by about 50% to about 3% as the acidification of the ocean shifts that over towards the ammonium side of the buffer. What this means is that there's going to be about half as much ammonia and about twice as much ammonium relative to today's values. This is significant because it's thought that Nitrosomonas actually uses ammonia, not ammonium, as the substrate to oxidize for the first step of nitrification. So probably nitrification is largely limited by substrate supply right now. Ammonium concentrations are very low in most parts of the ocean. But if we're going to decrease the available substrate by 50%, it's certainly possible that nitrification is going to decrease dramatically as well. There's not a lot of evidence yet on this. There's people working on it. But there is at least one paper that's been published looking at this. Next slide. This is a Huseman et al. Paper that was published in 2002. What they were trying to simulate was not the kind of ocean acidification we're talking about, but they were trying to simulate the actual disposal of waste CO2, say from a power plant in the ocean. So they looked at a very broad range of pH changes in incubations from two different depths and measured nitrification rates, and they went all the way down to pH 6. Hopefully, we're not going to passively acidify the ocean down that far. We're in real trouble if we do. But what's important is they saw an almost linear decrease in nitrification rates from pH 8 down to pH 6.5. Now, admittedly, they used rather coarse increments of 0.5 pH units, and they had some other issues with their work. For instance, they used very high saturating levels of substrate and so forth. But this does tend to support the idea that nitrification could be greatly inhibited by increasing acidification. acidification in the near future. Next slide, please. There is a paper, excuse me, that took these Huseman results and used them to build a model for the North Sea. This Blackford and Gilbert 2007 paper took this linear relationship plotted there on the left. They replotted that data I just showed you. And then they plugged it into a model, which the output is shown on the right. And what they're predicting is a reduction in nitrification of about 20% at 1,000 ppm CO2 would result in a change in the ratio of nitrate to total dissolved inorganic nitrogen of up to 10% in the North Sea. This certainly has implications for phytoplankton community structure. We know some groups, if you like to say it this way, prefer nitrate while others prefer reduced nitrogen compounds. So this could have big implications for shifts in phytoplankton communities. It certainly could also have big implications for the supply of nitrate reaching denitrifiers in the ocean in order to complete that cycle. And it suggests that if these are good predictions, nitrification could become sort of a bottleneck in the nitrogen cycle in the future. Next slide. If you take, there's not much in the literature on global average nitrification rates. There hasn't been enough measurements made to really get a nice ocean data view map of nitrification over the ocean yet. But Yule et al. In their paper in 2007 took what data they could find, and they calculated an ammonium-specific oxidation rate, a medium rate, of about 0.195 per day today. If you take that Huseman relationship and you simply apply it to that rate, it suggests that by the year 2100, the global median nitrification rate could decrease down as low as 0.17 per day, which is a significant decrease. All right, let's go on. How do you measure nitrification? Well, again, there's several ways to do it. I've given you a reference from Bess Ward, who wrote a really great review paper on this in Nitrogen in the Marine Environment, and that's a good place to start if you're interested in this. You can do it simply by measuring the changes in nitrite and ammonium concentrations in seawater. That's sort of a simple mass balance. This is an older way and not very sensitive. You can increase the information you get from this by using selective inhibitors of either ammonium oxidation or nitrite oxidation, which I've listed a few of those. You also can couple these inhibitors by measuring not the oxidation of nitrogen, but the reduction of CO2, that is the carbon fixation. So just like phytoplankton, you can use 14CO2 to measure nitrification rates in dark incubations and couple them with inhibitors if you like. The problems with both of these approaches is it's really hard to make sure that inhibitors are only affecting the process of interest. Many inhibitors have unforeseen artifacts, and all of these methods require relatively long, cumbersome incubations. So you can also use stable isotopes. You can use M15 nitrite or ammonium and with mass spec to measure nitrification rates. The general problem with this is because the levels of ammonium and nitrite are very low in the ocean, it's hard to reach true tracer levels, although we're getting better at that. You can also look at isotopic enrichment factors, since ammonium oxidizers produce isotopically enriched ammonium and depleted nitrite. Let's go on. The next slide, let's talk about silicon. Silicon is required element for a number of organisms that make shells in the ocean, diatoms in particular, but also silica flagellates and radiolarians. Most of the silicon in seawater is actually salicylic acid. It also forms a buffer system that has 2 pKa's, which I've shown here. But because those pKa's are so high, almost all of the silicon in seawater, dissolved silicon, is already protonated. So I wouldn't envision a large change in the chemical speciation of silicon over the next the foreseeable future from ocean acidification. It has a simple cycle with no gas phase or organic forms, unlike nitrogen. And so let's ask, how is silicon utilization affected by ocean acidification? Next slide. Most studies that have looked at this, and it's only been a few, have found no impact so far. So that graph on the left from Milligan et al. 2004 used a cultured diatom. They found a slight decrease in the silicon per cell from 100 to 370 ppm CO2, but no change from 370 to 750. And again, in that Berrigan mesocosm experiment, there was, on the right, there was no difference in silicate drawdown across 3 CO2 concentrations, suggesting that silicate utilization may not be directly affected by ocean acidification. Next slide. However, it could definitely be indirectly affected by changes in phytoplankton community structure resulting from global change effects. On the left is the Tortel et al. 2002 data that I've regraphed from that. They incubated equatorial Pacific phytoplankton at 150 and 750 ppm CO2, and they found a switch from Phaeocystis dominance at the low CO2 to diatoms at the high CO2. And sure enough, if you look at the y-axis, the ratio of silicate to nitrate used increased dramatically as diatoms became more dominant in the high CO2 treatment. So this is an example of an indirect effect on silicate utilization by CO2. The one on the right is from our North Atlantic paper that I showed you some calcification data earlier. We found no effect of CO2 on the silica to POC ratio of the community, but we did find that as the community shifted away from diatoms in the 2 high temperature treatments on the right, that sure enough the community became less silicified. So this is evidence that even though silicon isn't going to probably respond directly to CO2 changes, it certainly may respond indirectly. Next slide. One thing that Milligan found that was interesting is that empty silica frustules of diatoms dissolve much faster at 750 ppm than in 100 ppm CO2. So even though there was no effect on live diatom silicon requirements, no big effect, there was a big effect on the rate at which the frustules, such as might be sinking through the water column, dissolved. So this could have effects on the cycling of silicon in the ocean and change perhaps shoaling dissolved silicon profiles in the future. Next one. And then finally, silicon effects. This is a Bering Sea incubation we did a couple years ago. Again, the 4 treatments. Just look at the top panel, and you can see in the ambient control, the ratio of diatoms to non-silicified phytoplankton was about 2. But if you look at the 3 bars below that, green, red, and black, whether we raise CO2 temperature alone or together, we saw a decrease in the diatoms relative to the other phytoplankton, again suggesting an indirect effect on silicon utilization. Next slide. To measure silicon biogeochemical parameters, you can measure biogenic silica, or BSI. It's fairly simple. You just digest the BSI on your filters, the form silicic acid, and use a simple colorimetric nutrient analysis method. There's also a radiotracer, silicon-32, and I've shown you some silicon uptake curves there and some references. Silicon-32 is expensive and hard to obtain sometimes, but it is very useful if you can get it to do kinetics experiments with silicon uptake. And then finally, there's other methods at the bottom. I've shown you PDMPO which is a fluorescent analog for silicon, which is incorporated into diatom cells. And you can see those green glowing diatom cells are showing you where PDMPO has been incorporated into frustules in growing diatoms. So you can trace new silicon deposition that way. Next slide. Let's finish the major nutrients. Let's talk about phosphorus cycle. Phosphorus is required by all cells for nucleotides like ATP and nucleic acids, also for phospholipid membranes and some proteins. The big form of phosphorus in seawater is either inorganic orthophosphate or dissolved organic phosphorus. To consider just the inorganic phosphorus, at pH 8, it's mostly HPO4 2-, about 87%. But as the ocean becomes acidified, that's going to shift over towards H2PO4-. Nobody knows really what kind of implications that has for phosphorus bioavailability, if any. Again, phosphorus has a simple cycle with no gas phase, although organic forms are very important. And let's look at the phosphorus cycle in relation to ocean acidification. Next slide. The simple story, again, like silicon, is that the few studies that have looked at it have found little or no response to PCO2 in terms of the cellular phosphorus requirements of phytoplankton. So on the left, the phosphorus quotas of E. Hux across this 4-treatment experimental matrix. On the right, the Cyanobacterium synechococcus, with little or no effect of CO2 on phosphorus requirements. Next slide. Even though there's no effect, it's well worth measuring particulate organic phosphorus in your experiments because it gives you a normalizer for things that do change like POC, PON, and so forth. And actually it's kind of a coincidence because we use phosphorus as a normalizer in the Redfield ratio right now. So the fact that it doesn't appear to be very responsive to changes in acidification may be fortuitous for us to look at other elements. I've listed 3 methods here. Simple colorimetric POP method at the top. The radioisotope uptake method, we're using P33. You can use P32, but that's a lot nastier. 33 is much easier to use. And also, you need to consider that phosphorus actually does absorb on the surface of cells. And there's some literature on that I've listed at the bottom. The graph here is showing you P uptake rates using P33, and it's showing you the total cellular P in black, and the amount that's left after you use a surface wash technique to wash off surface-bound phosphorus in the open bars. So this is something you may want to consider if you're making phosphorus measurements, is this surface scavenging behavior of phosphate. Next slide. So let's take what we have been talking about in terms of carbon and major nutrients and put it together and look at effects on elemental stoichiometry. So I'm going to show you some data collections of C to N and N to P ratios from both culture studies using cyanobacteria and eukaryotic phytoplankton, and also from natural community manipulation experiments. Next slide. These are cultured cyanobacteria experiments that I've collected from the literature. Picocyanobacteria, not nitrogen fixers, on the left, and the nitrogen fixers, Crocosphera and Trichodesmium, on the right. C to N ratios are plotted in black, and N to P ratios are plotted in red. The 0 line means no change in these ratios from present day to future CO2 concentration. So those are delineated with circles. Those are the experiments in which no change in C to N or N to P was reported. And then a positive deviation from the 0 line means increase in the ratio, and a negative decrease would mean, from that 0 line would mean a decrease. What you can see right away is that for the cyanobacterial experiments that have been done, in every case, C to N and N to P ratios either stay the same or increase under high CO2 conditions. which seems to support that carbon overconsumption model that we talked about earlier. We go on to the next slide and look at cultured eukaryotic phytoplankton. We see a somewhat similar story. I've divided this up into culture experiments using diatoms, primnesiophytes, and flagellates of various sorts. And you can see in most cases, it looks very much like the case with cyanobacteria. A couple of exceptions where the C to N or N to P ratios of diatoms or primnesiophytes actually decreased at higher CO2. But for almost all the other studies, they either stayed the same or they increased. Go on to the next slide, please. These are natural community incubations, including a variety of shipboard, mesocosm, semi-continuous, continuous batch cultures using natural communities. And I've listed all the references and all the different environments they've been done in. And I think the first thing that jumps out at you is there is no consistent story for C to N or N to P ratios. If you look at the black C to N ratios out of the 11 data sets, C to N ratios actually only increased 3 times. They decreased 5 times and stayed the same 3 times. So I'm not sure this really supports that carbon overconsumption model as a general trend. And you can see the trends in N to P ratios using natural communities are very mixed as well. So I think we need to look a lot more closely at why the phytoplankton culture experiments are giving us a little bit different result than when we gather together the results of these mixed communities, which of course include bacteria, zooplankton, detritus, all these other things. There's a lot of reasons that they may give you different answer than cultures. But I do think we need to be cautious about deciding that C to N ratios are going to uniformly increase in the ocean at high CO2. Next slide. In terms of generalizing about elemental ratio effects, as I said, the C to N and N to P ratios of individual phytoplankton species that have been examined in culture often increase at high CO2 or stay the same. But if you look at natural communities that have been manipulated in CO2 experiments, they're a lot more variable. I think you need to be cautious when extrapolating from any one experiment to the whole ocean or to other regimes. We need to really look at this on a case-by-case basis. But what's obvious is there's a lot of room to do more work to understand the effects of ocean acidification on major elemental stoichiometry of phytoplankton and of other organisms in the ocean. So let's move on to the last section. I'm going to talk a little bit to wind up my lecture about trace metals and how they might respond to ocean acidification. Of course, the most important trace metal is iron. There's a whole lot of literature showing that it is a biolimiting micronutrient over maybe as much as half of the ocean's surface. Probably most of you are aware of that. The reason that we think iron might respond to increasing CO2 is because it's involved in a lot of processes that may be affected by acidification. Iron is required in large amounts for photosynthetic and respiratory electron transport, for nitrate uptake and reduction, and for nitrogen fixation. So iron needs to be looked at in the context of ocean acidification. It seems like an important thing to do. But it's not the only element that could be important. Other elements whose cycles that could be impacted by ocean acidification include molybdenum, which is also, along with iron, required for nitrogen fixers, as well as the so-called soft metals like zinc, cadmium, and cobalt. And the reason these might change is because all of these have been found by people like Francois Morel and Bill Sunda to be cofactors for various forms of carbonic anhydrase, meaning that the requirements for zinc, cadmium, and cobalt could potentially change as the availability of CO2 changes in seawater. The effects of acidification on trace metal biogeochemistry can include several things. First off, changes in biological requirements for trace metals. I'll show you a little data on that, what little there is. It also could affect their chemistry. Many of these trace metals, particularly the soft metals, their inorganic chemistry is dominated by carbonate complexation. Obviously, as carbonate ion concentrations go down with acidification, this could very greatly affect their inorganic chemistry. Many trace metals are also dominated by complexation by organic ligands produced by organisms. And what the effects of pH are going to be on the metal binding functional groups of these organic ligands and how that will change solubility and cycling of trace metals is a very active area of research right now. My lab is working on that, and Francois' lab is, several others. Next slide. So first, let's ask whether the biological demand for those metals that are used as cofactors in carbonic anhydrase, like zinc, cadmium, and cobalt, is going to be lower in the future high CO2 ocean. Remember, phytoplankton should have less need to synthesize carbonic anhydrase to obtain CO2 as CO2 goes up in the ocean. And so you might infer from this that they're going to require less zinc, cadmium, and cobalt as the ocean acidifies. What is the evidence we have for this? Well, there's only a little so far, but it does tend to support this. Go to the next slide. This is a culture study by Bill Sunda. He grew a diatom at 2 pHs, and he measured carbonic anhydrase activity on the y-axis, and then he measured the cell zinc requirement on the x-axis as zinc to carbon ratio. And what you can see is at lower pH in the black squares there, which of course means that PCO2 is higher, they not only have less carbonic anhydrase activity, but they require a lot less zinc than cells that are grown at a high pH where CO2 is less available, the black triangles. They have much higher carbonic anhydrase enzymatic activity, and sure enough, they require a lot more zinc. So this does tend to support that idea that I just threw out, that these metals used in carbonic anhydrase will be required in lesser amounts by phytoplankton as CO2 goes up. Next slide. This is a natural community incubation, one of the very first that was done that hasn't been cited much, should be more. Jay Cullen did this during his graduate work with Rob Sherrill off the California coast. They incubated a diatom-dominated community across a range of CO2 on the x-axis from 100 to 800 ppm, and they measured the cadmium to phosphorus ratios of the phytoplankton in 2 size classes. And you can see whether you look at the small phytoplankton in black bars or the open bars, which are the large diatoms, the amount of cadmium they require tends to decrease as CO2 goes up. And again, this could be thought to be consistent with a model where cadmium is less required for things like carbonic anhydrase at higher CO2 levels, although that's not the explanation they provided in their paper. So I'll let you, if you're interested, you may read that and see what it has to do with iron limitation is their idea. But it does tend to show the same trend that I was predicting, in that the amount of zinc and cadmium required by phytoplankton may decrease in the future ocean as it acidifies. So let's go back and talk about a whole suite of metals in the next slide. This is an incubation we did in the Ross Sea with a phascistus bloom, incubated at 380 and 750 ppm CO2. We then measured using ICP-MS a number of trace metals, and we normalized them to phosphorus. And what you can see right away is in this phascistus bloom, as we incubated it at high PCO2, the amount of cadmium, cobalt, zinc, iron, and manganese in the cells decreased as CO2 increased. So this is interesting in that it tends to support that they require less cadmium, cobalt, and zinc, whether for carbonic anhydrase or other reasons. But it's also a bit unexpected in that they seem to require less iron and manganese, which, as far as we know, are not directly involved in CO2 acquisition. However, we see a different story for iron if we look at nitrogen fixers. Let's look at the next slide. This is from that Fu et al. Paper. I showed you the data on the left a few minutes ago, where nitrogen fixation rates increased with CO2 in iron-replete cultures, but not iron-limited cultures. But look at the panels on the right. In the same experiment, we measured the cellular iron quotas of these cells. And we found that they mirrored exactly the trends in nitrogen fixation. So in the top right panel, the amount of iron in the cells increases as CO2 goes up, and it's directly proportional to the increase in nitrogen fixation rates, which makes sense since iron is required in really large amounts to make the nitrogenase enzyme. But it's a very linear relationship with, in terms of increasing CO2, increasing the iron requirement. However, in the iron-limited cells at the bottom, just as nitrogen fixation was not affected by CO2 under iron limitation, neither was the iron requirement of the cells. So this is a very different response from the photosynthesis response, where iron seemed to decrease in the cells at high CO2. In this case, when high CO2 stimulates nitrogen fixers, they require more iron, which makes perfect sense. You go on to the next slide. They also seem to require more molybdenum. Same experiment. There's not much difference in the molybdenum quota of the cells at 190 and 380 ppm, but it's much higher at 750. And again, this is where nitrogen fixation is increasing, and molybdenum is also the other cofactor for nitrogenase along with iron. So again, this tends to follow along with the physiological responses that we're seeing in our cultures of these nitrogen fixers. Next slide. This is trichodesmium iron requirements at 2 CO2s in the top slide. So you can see as we increase the CO2 concentration at which trichodesmium was grown from 370 to 750, we saw about a 40% increase in the amount of iron in the cells. And again, that mirrors very closely the amount of increase in the nitrogen fixation rate that we saw. Interestingly enough, if you take the same culture and you increase the temperature by 4 degrees at the bottom and leave CO2 alone, you see almost an identical magnitude of decrease in the iron requirement. So this kind of suggests an antagonistic effect between CO2 and temperature in the future, where it's possible that higher CO2 would tend to increase the iron requirement of trichodesmium, while at the same time warmer temperatures decrease the requirement for iron. Perhaps you end up with a zero-sum game, and their requirements actually are not going to change. And again, this illustrates the really important point that you have to look at other factors than just acidification if you really want to make more robust predictions of future trends. Next slide. Just to wind up with some methods for trace metal biogeochemical analyses. For the approaches, we can use both laboratory culture studies and field incubations and measurements. I showed you some examples of both of those. They're kind of tricky because what you have to do, you have to combine the methods for manipulating carbonate chemistry that you're learning in this course, such as bubbling with CO2 and so forth. At the same time, you have to keep your incubations scrupulously trace metal clean, because putting things like bubbling apparatus into your trace metal clean water is likely to contaminate your experiment and give you completely erroneous results. And so they are difficult because you have to combine ocean acidification methodology, such as this course is covering, with trace metal clean techniques, which you can read about in the Bruin paper that I referenced here. In terms of analyses for trace metals, there are radio tracers for many of these metals of interest. Iron has 2 commercially available radioisotopes. There are also isotopes for zinc, cadmium, cobalt, etc. Again, like phosphate, even more so, it's important to differentiate between metals that are just absorbed on the surface of cells and those that are in the cells, true biological metals that are being used in the physiologically. And there's the titanium and oxalate washes that can be used to remove these surface scavenged metals and differentiate between these 2 pools. To measure metals in phytoplankton or other organisms, you can use graphite furnace, atomic absorption spectrometry. It's an older method, still used in some labs, gives you 1 element at a time, can give you very sensitive measurements of trace metal levels. But most labs now are using inductively coupled plasma mass spectrometry, or IPPMS. You can do both measurements like you can with GFAAS. The advantage being is you get a whole suite of metals from ICP-MS instead of just 1 at a time. But most labs have switched over to this. There's also some really cool ways to couple ICP-MS with other instruments. For instance, you can use an HPLC in front of the ICP-MS to potentially look at trace metal speciation in your samples. You can also measure. Now there's growing literature on measuring things like stable isotopes of iron using multi-collector, or MC ICP-MS. So there's a lot of applications to trace metal biogeochemistry that could be applied to ocean acidification studies, and I've given you some references. And then finally, I've listed a really neat method, synchrotron X-ray fluorescence. It's been done by a few oceanographers, including Ben Twining and Phoebe Lamb, also have done some there at Hui. And I think this takes the prize as the most expensive analytical instrument ever invented for oceanography. It covers about a city block, and the only ones available are at the U.S. National Labs here. And they cost, I don't know how many billions of dollars. But you can get beam time if you apply for it. And what this allows you to do is measure the total amounts and even the localized at a subcellular level trace metals in individual cells and particles. So you can image these cells, and you can measure, for instance, how much iron, zinc, and so forth is in each individual cell. Next slide. I just threw this in to show you that there have been intercomparisons between these different methods of looking at trace metal quotas. This is one from the SOFEX experiment by Ben Twining, in which he compared iron to carbon ratios using radioisotope methods with the synchrotron methods. As you can see, in some cases they match up nicely, in others they don't. And we're actually involved in making some similar intercomparisons right now. Let's go to the conclusion slide. In conclusion, it seems that changes in acidification is going to really change the present-day ocean biogeochemistry of carbon and nitrogen. It seems like there's no way they can avoid reacting to this huge perturbation. It also seems from our limited information that there's not as big a response from phosphorus or silicon cycles, although they certainly may change indirectly in response to these other changes in other elements and to species composition changes in the ocean. The responses of trace metal biogeochemistry are just now beginning to be investigated, so it's hard to generalize. Second point is one I hope I've gotten across. You need to look at other global change variables if you want to get a complete picture. The ocean's warming, stratifying, nutrient and light supplies are changing, and the interactions of these variables with CO2 are at least as important to consider as ocean acidification effects in isolation. And this goes for all organisms, animals, plants, bacteria. I believe that using a only reductionist CO2-centric type experiment is not going to be able to give you a complete picture of what's going to happen in the future. You need to look at interactive effects. To do this, we need to build a new generation of studies, both experimental, observational, and modeling, that can address long-term changes in biogeochemical cycles. And they need to take into account things like the effects of acclimation, adaptation, and evolution of organisms in response to global change. And I think Deborah is going to talk to you a little bit about that in her lecture later this week. Finally, what I want to leave you with is don't get the idea that it's all been done. As a matter of fact, the responses of a lot of these key biogeochemical processes in the ocean have only been looked at very superficially or not at all. So there's a lot of major processes, things like anammox, things like assimilation, things like organic nutrient biogeochemistry that just have not even been looked at. And this leaves a lot of room for young investigators like most of you there in the room to work in this field. There's plenty of work for everyone. If we go to the next slide, I just wanted to acknowledge my students, my postdocs, and my collaborators on some of the work I presented, and of course the funding agencies that made it all possible. Thank you. Well, we're just about out of time. It's up to you, Heather, whether you want to take time for questions or not.
We'll take time for a couple of questions if people have them. I think they all want to go to lunch, Dave.
Well, go to lunch.